Phylip is a common software tool available for construction of phylogenetic trees. What is Phylip? Phylip stands for phylogeny inference. What are different characteristics? Basically, it is a software tool which is freely available, of course, and different versions are available for Windows, Apple Macintosh operating system, and Linux-based machines. It is basically one of the most widely used softwares for phylogenetic analysis. Of course, the other one is POP. And similarly, what are different parameters? We can use maximum parsimony tree, distance-based method, consensus tree, and bootstrapping for inferencing or evaluation of our trees. So what is data which should be input in a Phylip? It should be a molecular data which is from our macromolecules. It can be gene frequencies. Gene frequencies means number of occurrences of genes across sequences. Then restriction sites and fragments. What are those restriction sites and fragments? These are different groups of sequences. And then distance matrices and discrete characters within individual sequences. अब जो हमारे पास फाइलिप का जो सॉफ्टवेयर है उसमें रफली हमारे पास 35 डिफरेंट सेट ऑफ प्रोग्राम्स हैं विच आर रिटर्न यूजिंग सी क्लासिकल प्रोग्रामिंग और वो डिफरेंट प्रोग्राम्स क्या हैं वो भी हम देखेंगे और दे आर मोस्टली इन द फॉर्म ऑफ एग्जीक्यूटेबल फाइल्स विच डोंट नीड एनी अदर थिंग दे आर जस्ट execute on our machines as simple exe files. Moving on, what is the concept or what is the input procedure and then what is the output procedure or what is the format? For input, files should be usually in ASCII files or simple text files. Then what will happen? It will perform cluster W algorithm, which is multiple sequence alignment algorithm to align those sequences within each other. And then what will happen? These programs are stored in the file which is called in file format. And then we have output. Output would be written in out file format or tree file out tree format. Out tree format is basically hierarchical representation in the form of trees, and out tree is our textual or ASCII file format. Now this file is basically it is very analogous or comparable to another software POP, which has at the moment highest number of published trees which are related to phylogenetic analysis. Moving on, what are different usefulness of Phylip? Number one usefulness is that there are a high variety or high number of people who are very well familiar with this Phylip, so they can easily use that one. Then what will happen? This is very easy to use because we have just command line things using C programming, of course. But just we send commands and our results are outputted. Then there is a huge support of this Phylip software or Phylip commands for in other softwares also, such as for cluster, tree view, and other softwares. And finally, it is very easy to understand and easy to operate. This software has a very simple interface where we can put our queries and get our results. So what are different output mechanisms or visualization parameters? One was our out file, which was in text format, and the other one was tree view. In tree view, what we have? So in tree view, we have different options such as cladograms, then rectangle cladograms, radial tree, polygons, and etc. And this information, it will be graphically and hierarchically. In this figure, I have just shown some examples of trees which can be made by this Phylip program, and these are basically shown on different panels. That okay, this one is straight on the left hand side, then we have predicate type thing, then we have a scattered chart. Moving on, now I will discuss some of the important programs. As I said earlier, roughly speaking, वहाँ पर हमारे पास 35 program हैं, 35 programs which are available in Phylip. Some of them are given here. For example, number one is DNA parse. DNA parse basically it gives information of phylogenies which are based on DNA sequences, and it is presented by maximum parsimony method. Then DNA ML. DNA ML again similar to DNA parse, but the difference is using it is based on molecular clock or maximum likelihood based method. DNA MLK, it is phylogenies from NS using a maximum likelihood. Addition is only it will have a constrained molecular clock information. And then we have DNA distance matrix. DNA distance method is used to calculate distance between different sequences using 4 different distances. And similarly, we have some other method such as neighbor. Neighbor is used for finding neighbor join method between different provided sequences. We have prot parse. Prot parse is basically for protein sequence alignment, and again it uses parsimony trees. Then we have sequence boot. Sequence boot basically use bootstrap databases for DNA alignment, And then we have consensus trees. Consensus trees basically they are used for summarizing the result of different phylogenetic analysis outputs.